I've made a few notes to myself. I was able to get some background from the internet on you, and maybe just say a few words about your background.
So my background, I went to Harvard Radcliffe for as an undergraduate with a degree in biochemistry. I always had a interest in to become a physician. I was also very interested in research, and probably my greatest academic experience during those 4 years was as a biochemistry major, we were required to do a senior thesis. And so kind of a little bit unknowingly at the advice of a senior student, I stumbled into the laboratory of Walter Gilbert, Nobel laureate, who had a very thriving laboratory filled with undergraduates, so it was a very, very fun environment. We used to listen to Led Zeppelin and Carole King on the tape recorder while doing our experiments, and it was just a very, very fun environment, and I discovered that I really loved research. But at the same time, I was very clinically curious, and I knew in my heart at that moment in time that what I wanted to pursue was an MD.
Do you recall the title of your thesis?
It was, the subject was on characterization of memory B cells.
Wow.
Yes.
That's...
So it was a very nice system. Flow cytometry was new. And as an undergraduate, I had the opportunity to operate a flow cytometer on my lonesome. I was handed a laboratory manual, and I learned how to collimate a argon laser, and we were doing mouse injections and using 2-color, and at that time very advanced 3-color flow to characterize
Do you remember the instrument?
I believe, no, I do not.
Okay. I mean,
Coulter or probably a Coulter
Coulter or
instrument.
BD.
Yeah, one of the two.
Yeah, 2 and 3 color. Wow, that's great. Well, that would be an incredible introduction to.
I became interested. That was my introduction to immunology. That was my introduction to B cells. I subsequently went to Stanford for medical school, and I was delighted to meet the Herzenbergs, who had created these flow cytometry instruments. So I met Len and Lenore Herzenberg. But then I also felt like, because I was in medical school, I should try to figure out how to make this more clinically relevant. Ultimately, I worked with Carol Kleberger. I was interested in kind of the role of cytokines, and I had the opportunity for studying transplant rejection in the cardiac allograft setting with a new PI, Carol Kleberger. She was just terrific. So I was in medical school, but I also took 5 years through medical school. I had one dedicated research year that was funded by the Howard Hughes Medical Institute. And that was really also a life-changing experience. I wrote some of my first papers there. I learned how to more on molecular biology, so it was kind of a marriage of molecular biology together with immunology, and also being able to handle patient samples.
As I looked through your CV, a couple of things caught my eye. One was what I think now I understand is an early interest in what we call cell therapy, cell-mediated. Well, I think it was originally called cell-mediated immunology, and then it graduated to cell therapy. And you seem to have had an extensive experience with transplantation, and it sounds like not only cardiac but hematological.
Well, yeah. So I think when I was at Stanford, I thought I was going to become a rheumatologist. So I was busy learning all the manifestations of lupus and various autoimmune diseases. And this is the power of doing rotations through your educational experience. I came back to Boston for my internship at the Brigham and Women's Hospital. On my 3rd month, I happened to rotate on the stem cell transplant service, and I just loved it. So I learned about graft versus leukemia. I had never really seen any patients with transplant before, and the concept that the immune system could be harnessed to render cures was just captivating to me, and trying to understand the basis of why. So, and miraculously during that month, none of the patients that I saw died nor suffered intense graft versus host disease. So it just worked out that way. So I walked out with a very rosy feeling about the power of cell therapy.
They all had good graft versus leukemia effects.
It was manageable, and so I had a very strong, positive feeling about stem cell transplant. I was also really captivated by the idea that patients on the ward, some of them were enrolled in clinical trials, and the scientific rationale of those trials had only been discovered through preclinical data a few years before. So the idea of kind of translating preclinical scientific findings into tractable clinical trials where we could test a hypothesis, and then the idea that we could take those patient samples back into the laboratory and try to understand and feed forward into the next iteration of trials really captivated me.
How, what was the transition to CLL?
Yeah, so as you probably through looking at my CV, you realize that my postdoc was completely on chronic myeloid leukemia. And that was really at the time the stem cell, so my clinical training is in stem cell transplant. And back in the day, that's mostly the patients that we saw on the transplant ward. Then 2000 is when Gleevec happened, and that was really transformative for the landscape. So suddenly our transplant wards were not filled with patients with CML. That was around the time that I was transitioning into kind of the senior phases of my postdoc. And so if I wanted to study graft versus leukemia, I had to think about a different disease that was sensitive to graft versus leukemia, but not CML. And so by then, around a few years later, as an instructor, I wrote a clinical trial using whole tumor cell vaccines for patients with CLL. So these were patients who were getting stem cell transplant, and we were trying to use immune modulatory mechanisms to enhance immune reconstitution. We would collect CLL cells from patients. We would irradiate them. We would give them together with a cytokine adjuvant. And then we would immunize patients with whole tumor cell vaccines, autologous, back. So these were patients with very advanced disease.
And they would be irradiated but not fixed.
Right. They were basically dead cells,
Cells.
right? So that they wouldn't propagate back. And we were able to track really strong immune responses in those patients and track them out over time. The interesting piece there is that because we were collecting whole tumor cells, leukemia cells from these patients, and remember, these were CLL patients with quite advanced disease to have to go to transplant. Those were the cells that we had ready to extract DNA when genomic sequencing technology came along. So we recognized that across the river, over at the Whitehead, the Genome Center, that there was new sequencing technology coming along. And by then I was really interested in trying to use sequencing as a way to find tumor antigens. And those tumor antigens that would come from mutations, and maybe this could be the basis of another vaccine trial to enhance graft versus leukemia. Well, we had to test out this very new technology, and because I had all that CLL collected from our patients, that was the source of material that I learned how to do genomics together with my colleagues over at the Broad Institute.
Do we have, as of yet, a good antigen that's been generated by a CLL lymphocyte to be a tumor antigen?
There are a couple of interesting epitopes that we can go after, but I also want to say a word, which is that because we were treating advanced patients, the nature of the patient population that we were treating led to interesting genomic findings. So in other words, the first 6 patients that we studied 2 out of those 6 had a SF3B1 mutation. And because they had the same mutation, it mandated that we study it. And that kind of really led to our discoveries of some of the key recurrent mutations in CLL because of that interesting cohort that we were treating.
And the role of autologous transplant or even allogeneic transplant in CLL has perhaps gone the same way as CML?
Correct. I see patterns, which is actually progress for our patients. Yes.
One other thing that I made a mental note to myself was a while back, I think Dr. Kwok and you had a paper where you discussed the lymphocyte doubling time.
Yes.
And you described 3 patterns, the exponential and the logistic,
Yes.
and then indeterminate.
Yes.
And what exactly is the meaning of the word logistic?
It means that cells attain kind of a pattern in which they reach what we call carrying capacity. So there's a plateauing off. It's kind of a concept that's actually also in environmental science. So when you talk about kind of, I was hiking in the Grand Canyon and I talked to the guide, and her thesis was about the deer population in Arizona and how it relates to carrying capacity. So populations are either grow or are constrained based on resources. So it becomes a model for understanding how that kind of interaction happens.
The reason it caught my eye is that, well, the lymphocyte doubling time, I'm sure you're
Yes.
familiar with that. David Galton, I think, began that idea in his, I want to say, 1967 medical thesis, and talked about leukocyte or lymphocyte pattern, growth patterns. And he described the exponential, and he didn't use, he referred them as to type 1 and type 2. Type 1 is the exponential, and type 2 was what you're calling logistic, and he called that, I want to say, type 2A, and then there was a type 2B, and he described it as sawtooth. And I've never known what that meant, except just recently I saw a patient that had some immunological stuff going on, and it made me think that there is probably a lot of information in that sawtooth that
Yes.
that has at least escaped my understanding. And I knew that we would be having this interview, and I wanted to touch base with you on that. And also, you have a fellow in your lab that is Dr. Kwok, who's working on spontaneous remission.
I was going to say.
Yeah. And I told him something incorrect. I thought that Dr. Daniel Katowski was perhaps one of the earliest people to describe spontaneous remission. Galton has it in his thesis.
Oh.
I supposedly read it, but I forgot. So I thought you...
Those were there were 2 points that I would just make. The first is that, yes, in terms of the sawtooth, we do need to remember that there are a small proportion of patients who have spontaneous regression. We still don't understand it fully. I think we currently have the tools to kind of chip away at that question, and it's wonderful to work with Dr. Kwok on this really interesting question. And the second thing I would say is just that the study of exponential logistics puts our work kind of in a larger context of cancer evolution, and also in the realm of kind of the evolutionary mathematicians who have been thinking about this. So some of the terms that we use is actually borrowed from kind of the larger field and how the mathematicians think about it. So.
One last question. I noticed that you are a member of the National Academy of Science.
Yes, medicine.
Medicine. Okay. I don't think I've ever met anyone. I'm thrilled to. I'm probably being overly sentimental about this, but how did you do that?
I'm very honored
Yeah.
to have been elected to that August society. It's really a, a deep honor, and I think it's just, I think it's following our passions and really kind of keeping our eye on the bigger questions and just making progress.